The clinical validation of a SNP-based prenatal cell-free DNA screening test for fetal RHD status in a large U.S. cohort was accepted and presented as a late-breaking abstract at ACOG annual pregnancy meeting. Please allow me to share some background information, the study objective and methods, and then the data results and conclusions from that study. Non-invasive prenatal cell-free DNA screening for fetal RHD has the potential to guide Rh immune globulin administration during pregnancy for Rh-negative patients, and also to guide diagnostic testing and surveillance for alloimmunized pregnancies. The current standard of care for Rh-negative pregnant patients has essentially been unchanged over the past 30 years. In light of recent Rh immune globulin supply constraints, ACOG has highlighted that cell-free DNA testing for Rh is a reasonable option. Additionally, fetal RHD testing has been used in Europe for many years. The objective of this study was to assess the performance of Natera's next-generation sequencing-based cell-free DNA test for fetal RHD status in a large cohort of Rh-negative pregnant patients. Let's take a look at the study methods. Clinical cases with previous commercial SNP-based NIPT cell-free DNA testing for aneuploidy performing at Natera were selected. Maternal and fetal genotype were evaluated through prospective DNA analysis and then compared to routine serologic blood typing results of the newborn. The fetal RHD NIPT next-generation sequencing assay was designed to detect the presence or absence of the fetal RHD gene, as well as the non-functioning hybrid and pseudogenes with amplicons and probes dispersed along the 10 exons of the RHD gene. This is the largest clinical validation of fetal RHD NIPT with serologic newborn data performed in the United States. Table 1 reviews the study demographics and clinical characteristics. The cohort was quite representative of the distribution of Rh-negative status in the U.S. Maternal race was reported as 74% white, 7% Black, and 2.1% Asian. Overall in the U.S., the incidence of Rh-negative status is 15% in the white population, 5% in the Black population, and less than 1% in the Asian population. In this cohort, the average gestational age was 12 weeks, with a fetal fraction of 8.0%. These characteristics would suggest that this test has the ability to overcome some of the limitations of early European approaches, and that includes the need to be done later in pregnancy and lack of data on a diverse group of patients. This cohort includes a diverse mix of patients with an average gestational age of 12 weeks. The platform uses fetal RHD, uses SNPs to determine fetal fraction. Now on to the results and test performance. Figure 2 provides the data on assay performance. 655 samples had prenatal cell-free DNA for RHD genotype. There were 2 samples that received a no result, resulting in a no result rate of approximately 0.3%. There were 356 true Rh positives and 0 false negative results, resulting in an assay sensitivity of 100%. The specificity for fetal RHD was 99.3%. There were 2 false positive cases identified, in which the RHD gene was identified as being present. It is likely the result of an uncommon RHD gene variant, and the newborn serology showed an Rh-negative result. Of the fetuses with a negative RHD phenotype, we identified 3 with at least 1 pseudogene allele. The negative predictive value was 100%, and the positive predictive value 98%. Importantly, there were no false negative results. A false negative result could be catastrophic in clinical practice, resulting in sensitization if RhoGAM was withheld when the fetus is likely Rh positive, or in the case of alloimmunization, reduce surveillance when the fetus is actually at risk for hemolytic disease of the fetus and newborn. The test was optimized to reduce the risk of a false negative. In conclusion, the validation of fetal RHD in a diverse cohort and done early in pregnancy demonstrates accurate prediction of fetal Rh status. Importantly, there were no false negative results in the cohort, and the sensitivity was 100%. The large sample size provides confident results for clinicians. It also provides clinical utility. Early identification of fetal RHD genotype and status can assist patients and providers in the prevention and management of Rh alloimmunization. In a recent practice advisory addressing the current Rh immune globulin supply constraints, ACOG states that the use of NIPT to prioritize the use of RhoGAM and conserve its supply is a reasonable consideration in a practice setting that is experiencing shortages. Finally, fetal RHD testing has the potential to transform the clinical management of RHD negative pregnant patients by guiding Rh immune globulin administration for those at risk of alloimmunization, those carrying an Rh positive fetus, and by providing information to guide counseling and management as early as 9 weeks for patients that already have anti-D antibodies and can be done before the initiation of antibody titers and/or fetal monitoring. Thank you very much.